Hey everyone, over the last few videos, we have built our own tools to transcribe, translate, and parse faster files. But that was essential to understand the logic. But what if I told you that there is a professional library that does all of that and much more in a single line of code? So yeah, today we are introducing BioPython. It is the industry standard for bioinformatics, and learning it is like getting an exoskeleton for your coding skills. So let's get it installed and see what it can do. So first things first, we need to install the library. Since we are in a VS Code notebook, we can do this directly in a cell using pip. So we will install it like this. Pip install BioPython. Okay, so as you can see, BioPython library has been downloaded and successfully installed, and this is the version of the library along with NumPy. Okay, so once that is done, we can import the most important object in the library, the seq object. So in BioPython, we don't just use regular strings, we use the seq object. It looks like a string, but it has biological knowledge built in. Okay, so this is how we do it from bio.seq import seq. Then we will create a biopython sequence object. My DNA is equal to seq. Then we will create a simple string, and then we will print it. DNA sequence, my DNA. Okay, then we will see what happens. Okay, so now it has assigned this as the DNA sequence. Now, remember those functions we spent 10 minutes or more writing in the previous videos? Now, look, we will see how BioPython handles them now. Okay, so first is transcription. So, my RNA is equal to my DNA dot transcribe. Okay, now we will print it. RNA my RNA. Okay, then next is translation. My protein is equal to my DNA dot translate. Then we print it. Protein, my protein. Then next is reverse complement. My reverse complement is equal to my DNA dot reverse complement. Okay, then we print it. Reverse complement. Okay, now let's run this and see what happens. Okay, so transcription, translation, and reverse complement. Each is just one word. Not only is this faster to write, but it is also highly optimized for speed when dealing with millions of bases. So now you might ask, why not just use strings? So well, BioPython objects are smarter. So for example, if you try to transcribe a protein sequence by mistake, BioPython will warn you or handle the biological context correctly. It also handles the stop codons automatically. If you might remember, we use underscroll for stop codons, but BioPython uses an asterisk. Okay, so we will see what happens. Long DNA is equal to seq. Then we will create a string. like this. Then print long DNA dot translate. Okay, let's run this. Okay, do you see that asterisk at the end? That is the official biological notation for a stop codon. And BioPython follows international standards, ensuring your work is ready for publication or research. Finally, if you might remember that complex loop in the last lesson we wrote to parse multi-FASTA files. Well, BioPython has a module called seq.io, which is also sequence input output, that makes this very trivial. Okay, so we will import that from bio import seq.io. Okay, now we will parse a FASTA file in just one loop. For record in seq.io dot parse, we already have our genes.fasta file. Okay, and the format is FASTA. Okay, then print id is equal to record.id. Next, print sequence record.seq up to 20. We want to show the first 20 bases, right? And we can add some dots over here, right? After that, print length len of record. Now let's see what happens when we run this. Okay, with seq.io.parse, you don't have to worry about headers or new lines. It creates a record object that contains the ID, the name, and the sequence automatically. It is incredibly robust. Okay, so as you can see from the output. So today was a game changer. We moved from writing manual scripts to using BioPython, which is the powerhouse of biological computing. We also learned about the seq object and how to parse file using seq.io. Now that we have professional tools in our hands, we can start using real analysis. So in the next video, we are going to use BioPython to perform other tasks, and we will see that in the next video. Okay, so the notebook with all these codes will be on GitHub soon. So if you're enjoying switching to Pro Tools, please hit that like and subscribe button. And if there are any questions, please let me know in the comments. See you in the next video.